So ICER, first, we're an independent, not-for-profit organization, and our funding, as we like to keep updated on our website, shows that approximately 77% of our overall revenue comes from nonprofit foundations. We do have about 8%, as you'll see, from health plans and provider groups, and 13% from manufacturers that are contributions that go directly towards a policy summit program and other activities that are separate from our research activities and report creation. And we have a very small residual amount of funding from state governments for some of our work. So why are we here today to talk about this topic? Let me just start with a few quotes that we heard from patients and patient advocates. One was, as my MS specialist said, I have now moved out of the heavily funded relapsing remitting research category to the little known, least explored category of SPMS. I may have a rough road ahead. And another quote, actually from Kathleen, who's with us. Oh, no, she's not here today. Sorry. I think she is later. A quote that she gave us was, "Siponimod is an important approval and will hopefully stimulate important research. Price is an important factor in determining access to a medication. So while Mayzent, that's its trade name, is not priced at the top of the MS drug list, we believe the price is still too high." And she was speaking on behalf of the National MS Society. So in part from these kinds of quotes, you can see that we are dealing with an area in which there is significant unmet need. SPMS has not had a formally labeled treatment before, and siponimod was viewed as a potentially important new advance for patients in that spectrum of the condition. And anytime we have new drugs for an unmet need, it is going to raise questions about, which patients is it appropriate to start on these drugs? Should they try other drugs first and then move to this second? Those kinds of questions. And always, because it's new, there will be questions about whether it will add to the overall costs of care, decrease them, in what way. Meanwhile, in the background, we have employers who are important for many of us in employing us and in helping with provide health insurance. So they have to see the overall picture of the increases, if they are such, to the cost of care as they try to work with their employees to maintain an affordable health benefit structure. And we all know that patients can have difficulty accessing drugs. That can be because of their own out-of-pocket burden. It can be because of the rules of getting to a treatment that they and their doctor think is best for them, that does require them to try other things in different ways. And there are a variety of ways that they can face challenges that are wrapped up into the overall need to think about effectiveness and value. So that's why we are here today. We hope that there is a benefit to patients, but also to the whole health system if we look here and now at this treatment, but think more broadly about a structure in which the health system can move towards a place in which it rewards value in a way that fosters innovation while maintaining sustainability from an affordability perspective. So our report today hopefully contributes to that. Here's how it's developed. First, we start about 8 months ago with a scoping phase, and to us that means that we reach out to patients and patient groups, to the manufacturers, to clinical experts and their societies, and we say, what do we need to know? Tell us about the diversity of the patient experience. Tell us about the arc of innovation in this space. Tell us a lot about what the experts can tell us and what they can't, about what really matters to patients. And we take that as our guide and go and do an evidence analysis. And we look at the published literature for sure, but we try to seek out patient-reported outcomes, other data that might exist somewhat in the gray zone where it hasn't been formally published yet, but might be important for policymakers to know about. We work in collaboration often with outside academic investigators, and in this case we worked with Dr. Sharaf, whom you'll meet, and also a cost-effectiveness team from the University of Washington. Our work goes through several rounds of public comment and revision, and a formal expert review from 3 neurologists in this case and 2 different patient groups. Now, our reports are structured in a way to try to foster a specific approach to talking about value. We call it our value framework, so I want to share that briefly as well. We start with the idea that every analysis of value should have an explicit goal in mind, and for us, We try to describe it as a mixed goal that they have to support each other. A fair price, fair access, and future innovation. That the three are joined as goals for our efforts in review and in public discussion. Now, for many policymakers, and this is going to be at the population level, not at the individual patient-doctor level, but for policymakers, we think it's helpful for them to think of reaching that goal by focusing on the evidence on long-term value for money and short-term affordability. First, long-term value for money itself is a composite idea in which we think that we need to focus first on the comparative clinical effectiveness for patients. We do need to think about the long-term incremental cost effectiveness. We also, in our reports, and we'll talk about them today, want to make sure that we capture other benefits or disadvantages that might not be in the clinical studies, might not be measured at all, or it might be measured inadequately or incompletely. And so those are things, again, that could be very important to patients, their families, the health system. We want to try to bring those into consideration, as well as broader contextual considerations that often focus on some of the ethical and social values that should modulate, if you will, how we look at evidence and making decisions. And even if we do have good long-term value for money, decision makers do need to think about the short-term affordability. So there are reports focus a bit on potential budget impact analysis. Those tend not to be such a direct part of the voting or the issues that we talk about today, but it's always important to think about for decision making, and it will, in some cases, be a major issue for certain drugs. So that's the overall structure of the report, and in some ways it even models the way that we'll be talking about the information at the meeting today. The agenda for this morning, in which we'll do this topic before shifting to another topic after lunch, is about an hour of discussion, presentation and discussion of the clinical and economic information from the report with questions from the CEPAC. We will have a phase of manufacturer comment and discussion, public comment and discussion, followed by the briefest of possible breaks to let people stretch their legs, and then we'll come back and do further discussion and deliberation among the CEPAC with clinical experts and patients before they vote on effectiveness and value. Then we'll have an abbreviated discussion of some key policy points. That's when we'll talk a little bit more about how we might envision translating the available evidence into recommendations for future research, but also into how payers, especially in some ways, how they might think about translating it into coverage policies and into pricing negotiations. So we'll try to touch on some of those points with the topic this morning before breaking for lunch. I'd like to introduce the folks at the front table here who will be available to the CEPAC during the morning and then play an important role in the afternoon as well. Sorry, not in the afternoon, but in the latter part of this morning as we talk about the policy aspects. Actually, why don't I just invite you to briefly introduce yourselves, if you don't mind. And any potential conflicts of interest are mentioned up here, but if you have anything specific to add or to say about that, please do. Dr. Cohen?
Good morning. Good morning.
I think you have to press it continuously.
Oh, okay. Good morning. My name is Bruce Cohen. I'm a professor of neurology here at Northwestern University in Founder School of Medicine, and I'm director of the Northwestern Comprehensive MS program.
Hi, I'm Annetta Langer-Gould. I'm the lead for clinical and translational neuroscience at Southern California Kaiser Permanente. I'm an MS specialist and an epidemiologist.
Good morning. My name is Ann Moore, and I have been asked to be here by June Halper with the Consortium of MS Nurses, and I'm a patient advocate. I myself have MS and have had it for 30 years.
Hi, I'm Holly Schmidt. I'm with the Accelerated Cure Project for Multiple Sclerosis. I'm the Vice President of Scientific Operation, and we're a research-focused patient advocacy organization in MS.
All right. Thank you very much for being here, all of you. I think you've all had to travel at least some distance, so we appreciate your input today and throughout the process. All right, with that, I'd like to introduce Dr. Ravi Sharaf, who will be presenting the information on comparative clinical effectiveness, and we'll kick off from there. Thank you.
Thank you, Steve. It's a pleasure to be here. So the evidence team consisted of myself, Patty Sinott, Noemi Floich, and Serena Heron-Smith, and none of us have disclosures that are relevant to the presentation today. This project assesses the comparative clinical effectiveness and economic impact of siponimod in the treatment of secondary progressive MS. I'll be talking about the evidence review now, and then Dr. Lisa Blodek, a research associate at the University of Washington, will be discussing the economic analysis. Multiple sclerosis is a chronic, inflammatory, demyelinating, and neurodegenerative disease of the central nervous system. Up to 1 million people in the U.S. are affected, and annual disease-related costs are about $24 billion and are estimated to increase as prescription prices outpace inflation. We'll verbally talk about MS nomenclature and then hopefully reinforce it pictorially in a second. But relapsing remitting MS, or RRMS, is the initial presentation in 85 to 90% of MS patients. Relapses are the episodic development of neurologic symptoms that may resolve, and if they do not resolve completely, you get the accumulation of disability and potentially progressive disability. Relapses usually reflect MRI-detectable CNS inflammation or lesions. One thing to note is that the nomenclature can get a little bit tricky here, and so there's a difference between disability progression and progressive disease, which we'll talk about next. Progressive MS is characterized by increasing neurologic disability that occurs independent of or in the absence of relapse, and that's distinct from RRMS. Progressive disease can be active in the presence of clinical relapse or MRI findings that are consistent with MS, and this active, not active distinction is important for today's discussion. Patients have primary progressive MS when they have a progressive course from disease onset, and they have secondary progressive MS when they start with RRMS and then progress to SPMS. So pictorially, let me see if my pointer doesn't work. Let me see if this guy works. There we go. Okay, great. So this is a typical disease course of a patient with RRMS who transitions to SPMS, though that does not happen in all patients. So if you can focus on this green line, these vertical boxes are episodes of relapse, which sometimes resolve completely and sometimes don't. And so if patients don't get back to their horizontal green baseline, they accumulate disability in this stepwise fashion. You'll note that relapses are often, but not always, associated with MRI activity, which is down here. In some patients, RRMS can transition to SPMS, which is secondary progressive multiple sclerosis. What you'll note is SPMS is characterized by decreased relapses here, decreased MRI activity, but increase in clinical disability. So distinguishing the phenotypes can be hard because there is overlap, there is no biomarker, and SPMS is a diagnosis that's made retrospectively. To understand the impact of SPMS on patients, it's best to hear it from patients themselves. So ICER was fortunate enough to be part of a survey that the MS Coalition did with about 2,800 SPMS patients, and these are some of the quotations that came from the survey. I lost the ability to stand, transfer, or walk a few steps with a walker in 2010, which had a huge impact on my life. My cognitive dysfunction has continued to become more impaired. I thought I would be working outside of my home by now, but my options are really limited. Because of this, our finances are tighter than I thought they'd be. My kids know that I can't do what a lot of other moms can do. My husband has to do so much more because of it. I've given up thinking that there is anything out there to help me. So the therapeutic goal in MS is to decrease disease activity and disability progression. And as Steve was mentioning, when we started this review in August 2018, the landscape was that there was multiple disease-modifying therapies that were approved for relapsing forms of MS. And this can get a little bit tricky. So RRMS is a relapsing form of MS. At that time, in August 2018, active SPMS, over here, was not explicitly defined by the FDA to be a relapsing form of MS, but medications used in relapsing forms of MS were often used in active SPMS. The only medications that had FDA approval for progressive MS were two, and so one was ocrelizumab here, which is used in PPMS, and the other was mitoxantrone in SPMS, although mitoxantrone was seldom used because of toxicity. Ocrelizumab was used, we heard from our stakeholders, in SPMS off-label. So when the label for siponimod came out with a potential indication for both non-active and active SPMS, that was of considerable interest to us and I think stakeholders in general. Siponimod is an oral selective sphingosine 1-phosphate receptor modulator. It has anti-inflammatory activity. The mechanism of action is similar but not identical to fingolimod, which is an approved FDA medication for relapsing MS. When the FDA did review siponimod in March of 2019, a couple things happened. And so it was explicitly approved only for use in relapsing MS. But the FDA now stated that active SPMS was part of relapsing forms of disease. Siponimod was not approved for use in not active SPMS. And the FDA also clarified that DMTs, that medications that could be used in relapsing MS, were also approved for use in active SPMS, which meant that siponimod was now one of many medications that could be used in active SPMS. After that label, we went to our stakeholders and talked about the implications of the label, and the consensus, I think, was that an evidence review of siponimod in a not active SPMS population was still of interest, and so we decided to continue with the review. Briefly, for the evidence review portion, we conducted a systematic review following PRISMA guidelines. Our comparators were informed by our stakeholders and were basically treatments that had some efficacy in SPMS or were commonly used in practice regardless of FDA indication. They included best supportive care, ocrelizumab, beta interferons, and natalizumab. For the presentation today, we talk about siponimod and ocrelizumab. These are some of the outcomes that we gleaned from the literature, highlighting the expanded disability status scale, the EDSS. So it's the oldest, the most commonly used validated measure of disability. There are caveats to its use and measurement. 0 is somebody with MS who has completely normal function, whilst 10 reflects death from MS. And in the middle is 5 is ambulation with a cane. It's an index that's pretty weighted on mobility and lower extremity mobility. In the EXPAND trial, the primary outcome was, which is a trial of siponimod, the primary outcome was confirmed disability progression at 3 months, which for them was a clinically significant progression in disability by the EDSS scale that persisted for 3 months. There also was a timed 25-foot walk test, which is the average time it takes for a patient to do 2 25-minute walks. We also collected data on relapse, though we don't present that data in this presentation today. From our literature search, we identified one phase 3 trial, EXPAND, of siponimod versus placebo in SPMS patients. There was no head-to-head studies of siponimod versus an active comparator. There was 2 studies of ocrelizumab, though they were not in SPMS populations. We tried, but felt that we were unable to perform a network meta-analysis due to differences in study eligibility criteria, enrolled patient demographics, and study endpoints. And Novartis did supply us with a matched adjusted indirect comparison, which we'll be talking about a little bit later during the economic analysis section. The EXPAND trial of siponimod was a multinational, multi-site, double-blind, randomized controlled trial of siponimod versus placebo. There were some FDA some concerns raised by the FDA during their review. They noted a possibility of compromised blinding in the trial, and also noted the possibility of misclassification of patients, meaning that they felt that some patients represented a late RRMS population versus an SPMS population. Results from the trial are significant as follows, and so there was decreased disease activity in the overall SPMS population, and there was also decreased disease progression as measured by the CDP3 in the overall SPMS population. But you'll note that this effect was more pronounced in those with active disease compared to those with non-active disease. For those with non-active disease, the confidence interval did cross 1. We are cautious with interpretation of subgroup data. I think the thinking was, given siponimod's mechanism of action, it certainly, which is anti-inflammatory, it certainly was plausible that it would have a greater effect in patients who were active versus non-active. One other thing to note is that there was a discordance in disability in mobility outcomes. While there was significance found on the CDP3, there was no significant effect found on a timed 25-foot walk test. So in order to further elucidate whether siponimod had activity in not active SPMS patients, there were a couple post-hoc analyses that were done. And these were post-hoc analyses that were, this slide, our post-hoc analyses that were done by the EXPAND investigators. And so they basically used 3 methods, a principal stratum analysis, as well as 2 hypothetical strategies to look at this question of whether siponimod affected progression independent of on-study relapse activity. And the bottom line was that the estimated CDP from these analyses ranged from 0.8 to 0.86, though they were only significant in one of the 3 analyses that were performed. The FDA also did their own post-hoc analyses to look at the effect of siponimod in non-active SPMS. This is an analysis that was done in patients that had had no relapses 2 years prior to study start or during the study, and there was no significant difference that they found. And so their interpretation, given this data and given other subgroup analyses which they did, were that the pivotal trial results provide insufficient evidence to support a claim that siponimod is effective in patients with SPMS who are not continuing to have relapses or in patients with not active SPMS. Regarding adverse events, most adverse events were not medically serious and were treatable or reversible. Treatment was continued in about 8% of siponimod patients, about 5% of placebo, and the indications for discontinuation are there. Shifting briefly to ocrelizumab, which we looked into because we heard from our stakeholders that it was one of the more commonly used medications in SPMS. We did not find any primary data in SPMS for ocrelizumab. The 2 trials that we did find were either in a PPMS population or a relapsing MS population, and both showed significant reduction in CDP3. So broadly speaking, talking about controversies and uncertainties in this field, there is a lack of comparative effectiveness data in SPMS. In the EXPAND trial, there was a discrepancy between mobility outcomes. The fundamental question is, does siponimod work in non-active SPMS? Does it work independent of its effect on disease relapse? And per the FDA's assessment, their conclusion was that there was insufficient evidence for the EXPAND investigators from their post-hoc analyses. They noted that there was an effect that was confirmed. The FDA also had some other concerns. There was noted to be dropout after relapse in the placebo arm of about 8% versus 3% in siponimod, but the absolute risk reduction from the medication in the overall population was about 5.5%, and so that was a concern. And then, as mentioned, there was notification of potential issues with blinding and misclassification. Other benefits to be considered in this realm. SPMS has a high lifetime burden of illness. There are few medications with data to support its use. A delay in disease progression and activity may improve patient-centered outcomes and caregiver burden, and siponimod is oral, whereas the other medications that are used tend to be sub-Q, IM, IV. Some comments that we received, some stakeholders felt that ICER should discontinue the review after the FDA label came out. Again, it was what we heard from other stakeholders about the desire to look at the evidence for non-active populations, which made us continue. So in summary, SPMS is a devastating disease that impacts patients and their caregivers. Siponimod reduces relapses and has manageable harms in active SPMS, but there's uncertainty regarding siponimod's benefit on progression independent of relapses, meaning its effect in patients with not active SPMS. So for our evidence ratings, for active SPMS, we gave siponimod a B+, meaning that we have a high certainty that siponimod provided at least a small net health benefit in patients with active SPMS. We felt that there was insufficient data for the other two recommendations. We had low certainty about the net health benefit of siponimod versus best supportive care in patients with non-active SPMS, and we had insufficient data to conclude that the benefit of siponimod was better or worse than any of the other comparators used. Okay.
Could you, as one of the non-clinical people at the table, could you go back to slide 18? I want to make sure I'm understanding active versus non-active SPMS.
Yep.
So tell me if I'm looking at the right thing. The green line in SPMS is active when it sort of goes up. And even though it's continuing to go up in sort of a steady, that's the non-active, is in between the...
I can give it a shot.
So I think the figure's a little misleading. This is the kind of figure that pharma uses a lot to describe the disease course. In reality, that second relapse you see there in that course, that usually never happens. So there's a period in transition where they are still at risk of relapse, but they have far fewer relapses. But over time, it's a purely progressive course, and it looks identical to primary progressive MS. So when the patients will say, "Why is my MRI stable when I'm getting worse?" You examine them; they truly are a little bit worse. Their life is changing, but it's not a relapse. It's just typical slow, insidious disease progression.
So the slow part of it getting worse, even... That's the non-active part. It's just slowly, but there's no...
There's not been a relapse. We use more generous definitions in Kaiser. We say the last 3 to 5 years. Sorry. We say the last, if they haven't had a relapse in the last 3 to 5 years, if they haven't had a new lesion on MRI. So we don't stick with just GAD positive. If we see some progression on MRI, it suggests that they still have active inflammation that's being driven by a peripheral process, and that they may still be amenable to immunotherapy.
Yeah, if I can comment, one of the things that's important to understand is that the MRI doesn't show us everything that's going on in MS, and that the MRI shows us the things that are going on in the domain of inflammatory MS occur about 10 times as frequently as symptoms change. So consequently, those little bars which represent attacks are just the tip of the iceberg. That the MRI lines which represent new active lesions are also just a portion of the iceberg. We know that there is both an inflammatory component and probably a non-inflammatory component going on as the secondary progressive MS changes. Okay, we know, for example, that there are inflammatory components in the meninges, that occur specifically in secondary progressive MS, and so that's the reason that we think some of these medications may work in secondary progressive MS.
So tell me if I'm oversimplifying. So is active when there is sort of active inflammation that you can see in it?
So we define active as the presence of inflammation, either in the form of relapses or accumulation of abnormalities on MRI.
The FDA defined it, sorry. In the FDA analyses, they're defining it as primarily as a relapse within the last 2 years. But they do point that out as a limitation because that's all that was collected, and would have said it would have been nice to know last 3 years. So.
I think it's also important to point out that the goal of disease-modifying therapies is to reduce inflammation, and most people with MS are, or at least they begin with DMTs and often switch, and there are many highly effective DMTs out there now. So it's impossible to say in a case where somebody's not having relapses or MRI activity, whether that's just their natural course of disease or whether their disease is being very well controlled by a DMT. And I think that's one of the flaws with the definition of active and non-active MS.
Tim.
Thanks. Thank you. Nice presentation. I have a question and a comment. With regard to relapse in the study and the absolute differences, was that a clinical definition or did that include imaging?
Yeah, so clinical.
It's all clinical.
So relapse is a clinical definition. If you're active, that's a combination of clinical relapse and or MRI indication of MS.
Okay.
They did do, however, an analysis of change in the volume of new MRI lesions, which did show a benefit. Okay, so they did indirectly pick up relapses that way as well.
Okay. No, I just wanted to ensure that it was a clinical rather than a radiographic. And then the comment, we talked about this a bit last night, but I still struggle with calling it a B+. We have a single randomized trial in a selected group of individuals that you yourself note some concerns about both risk of bias and other things. I think the trial suggests that there's some benefit. I have real difficulty saying that there is at least high certainty of at least a small net benefit. We have one trial. I propose that the evidence suggests that it is a C+, where we have more moderate certainty or at least high certainty of at least a comparable net health benefit. I just think it's hard to get to high certainty on one trial.
Tim, David.
Yeah, I'm happy to comment. Do I need to do something? There we go. Thanks. So I'm not going to argue hard for one of those ratings over the other. One of the things that happens, though, I've noticed a lot with our ratings, is that B+ sounds like it's almost an A. And it doesn't mean that. It means that we believe that we're pretty sure it has a small benefit, and it might have more than a small benefit. And here we're talking about active SPMS, which is a relapsing form, where multiple disease-modifying therapies have worked, where it decreased relapses, where a very similar drug, fingolimod, decreased relapses, and comparing it not to any of the other disease-modifying therapies, where we certainly wouldn't have said B+ against other disease-modifying therapies, but against no treatment. And the question is, how sure are you that if the only drug you had to give somebody who was having active relapsing disease with SPMS, that you ought to give them this rather than nothing? We thought we were pretty sure that you ought to give them this rather than nothing because it almost certainly has a small benefit. So that's where we're coming down on it, but we could imagine people coming down somewhere else. That's how we got to B+.
Thanks. That's good. Let me just, part of the reason I bring the comment up, it's not just a pedantic one, I don't think. but I think it sort of sets the stakes of how certain we are. And then, as Holly mentioned, part of her role is to advocate for additional research. And if we say that there's high certainty, there's very little likelihood that anybody is incentivized to do additional research beyond just the population that's there. I think the data are not sufficient to negate additional research, and I think additional research would be very valuable.
Tim, thank you. Gregory, did you have a question? Make sure you hold it down.
Yes, I have a question on the EXPAND trial results. We have a breakdown in slide 29 for active disease and non-active disease. And we're given hazard ratios, active disease 0.65, non-active disease 0.85. The upper bound for non-active disease is 1.05, so it overlaps 1. We're not given an upper bound for active disease, and we're not told whether this breakdown of active and non-active disease was a pre-specified subgroup analysis, or is this a post-hoc analysis?
So these analyses for active or not active by MRI outcomes were pre-specified. The confidence interval for active was about it was less than 1. It was in the high 0.9s. Yeah, 0.95. Yeah.
I'd like to comment a little further on that, because this is actually a central issue, both on commenters and discussed internally at ICER. Often you wouldn't do a subgroup analysis without a very good reason to argue that there's a difference going on here. And I think, for instance, the manufacturer would like to take the position, potentially, that that hazard ratio of 0.79, the primary outcome of the trial, applies to every patient with SPMS, active or non-active. We heard from various stakeholders up front, including Dr. Langer-Gould, that we should be prepared to separate out those numbers before we start looking at the data, because there was lots of a priori reason to believe that siponimod would work better in active disease than non-active disease. And so rather than the usual subgroup position of saying we should only do a subgroup analysis if there's convincing evidence that the subgroups are different, we were told there's convincing evidence that you should look at these subgroups differently before we looked at the data. And so that's a different way of thinking about a subgroup analysis, and it's important to consider in this situation for why we're not willing to say that for all patients with SPMS, there's a 21% reduction in disability progression. We don't think that that number applies to all the patients. We think it applies, we think there's more in the active disease and less or none in the non-active disease.
Reem, would you have a question or a comment?
So thanks for the presentation. I think my question is more related to the outcomes chosen in the trials. And clinically, these outcomes are geared more toward, the EDSS or the timed walk test. It's about walking activity, which my understanding as a clinician, that is not always how MS would progress or would show itself. So I would like to just hear a little bit about how does that correlate, or how are these outcomes actually representative of true progression of the disease?
I think I'll back up. So I'll answer that question and let other people comment, but I also back up. I think, I read the FDA report on the plane, and it's, I think they saw something in the trial that I saw when they first published the results. They didn't recruit primarily progressive patients, that there's some funny business going on in how they defined SPMS and requiring documentation for it, that it looks like largely they pulled in relapsing remitting MS patients, some who were at the transition to SPMS. So if you look at the inclusion criteria for the trial, It says they had to have disability progression in a couple years prior to entry, and they have to have documented secondary progressive MS. There was supposed to be a form, a physician attestation form, that was to be filled out and sent to a central agency where they review, a central review board that confirmed that they really had SPMS before allowing them into the trial. This funny business about the unblinding of the patients, that sent some, so thank God for the FDA. They sent the Office of Surveillance Epidemiology out to inspect these sites. And while they were there, they found out that not only were there only 7 to 10 patients where there was a breach in the blinding, there was like 101, and they couldn't find any of these attestation forms. And then it turns out there was never a form that was required to be filled out. Okay, so that's not something I knew before I read the report, but what you can see from the data in the report is that the progression rates are quite low. They match the terflunomide relapsing remitting trials and the pivotal Avonex relapsing remitting trials for this confirmed 3-month disability progression endpoint. They are far lower than what we saw in fingolimod PPMS, where approximately 50% of the patients had confirmed disability progression. So that already, just seeing those event rates and the amount of inflammation in these patients in the 2 years prior, how many of them had relapses, and how low their EDSS is at entry. So half of the patients, the median EDSS was a 6, which is a cane. Only cane and walker. Of course, they were in wheelchairs. They were not allowed into the study. But they also had patients that had only a 3.5. That's a very, very healthy SPMS patient population. We see that, but how they found so many, it only makes sense if what they really recruited were patients with active relapsing remitting disease. I think one of the arguments was, well, we did ask for that documentation of the EDSS progression. They do have that. That should have been enough. But that does not work if they have active relapsing remitting disease, because you can accumulate disability from incomplete recovery from attacks.
So if I can go back to your question, one of the drivers for choosing EDSS is that's the only endpoint that the FDA will accept for a licensing trial for a progressive disease agent. So that was mandatory. To the point that it doesn't reflect everything that's going on in MS, that's absolutely correct. So there are cognitive changes that go on in MS which are poorly reflected by the EDSS. There are upper extremity functional changes that go on in MS that are poorly reflected by the EDSS. We know that, and we use other scales to compensate for it. A second thing, and this was, of course, a choice of the pharmaceutical company, was the design of the study. The design of the study was a time-to-event endpoint. That endpoint was a 3-month change, which, as Annette pointed out, is similar to what we see in relapsing remitting studies, because you can get short-term disability change with relapses. Consequently, the nature of the design favors an increased proportion of individuals who have relapses during the study or have subclinical activity reaching that endpoint, as opposed to people who have indolent progressive disease, which would only be picked up by a longer study. So there is a little bit of a bias that's inherent in the design of the study, which is at play here as well.
Ravi, thank you very much for your presentation. It was excellent, and all the comments so far. We're going to move now to the economic modeling part. Aaron, if we can hold your question to the probably to the vote period, or possibly then this next session from Lisa. Thank you.
Hi, so I'm Lisa Bodek. I'm a research associate with the University of Washington, and we were tasked with the economic analysis for siponimod. I'd like to recognize Josh Carlson, who's here at the University of Washington, and also the ICER team with Sume and Rick. And also acknowledge all of the comments we've received on the draft version that helped us refine this model. And we have no relevant disclosures. So our objective was to estimate the cost effectiveness of siponimod for the treatment of SPMS, and we looked at both the overall SPMS population as reflected in the trials, and also the subpopulation with active SPMS. And as we've heard, there's many ways to define active SPMS, given the data that we had and how it was pre-specified within the trial. We used the definition of evidence of relapses within 2 years of enrollment as a proxy for our active SPMS population. methods. We used a Markov model from the United States healthcare sector perspective with a lifetime time horizon, discounting of 3% per year for costs and outcomes, a cycle length of 1 year, and a few different primary outcomes. So we looked at the cost per life year gained, the cost per life year of ambulation gained, and the cost per quality gained. Our health states were based on EDSS. This structure with Markov model based on health states of EDSS has been used in many models of multiple sclerosis, both relapsing, remitting, and secondary progressive. And you can see it ties to both the clinical trial endpoint with confirmed disability based on EDSS and a change in patient's health state. It is biased towards lower limb ambulation, but that's what we have to work with. With the cost per life year of ambulation gain, that ties back to EDSS. Beyond 7, it's defined as essentially restricted to a wheelchair. So we considered life years in states above 7 to be a life year of ambulation. Each EDSS health state has associated with it a risk of relapse, a health utility value, a risk of mortality and direct costs, also indirect costs that we considered in a scenario analysis. We first modeled out the natural history of SPMS based on an established cohort. The siponimod efficacy was applied to this cohort. Once patients discontinued siponimod, they were assumed to follow the natural history transitions. We employed a few stopping rules. For the overall SPMS population, there was no stopping rule. Currently, there's no guidance or precedent for when you might stop an SPMS patient without active disease on one of these drugs. So for the overall SPMS population, we assumed they would remain on siponimod, unless they discontinued for some other reason based on the clinical trial discontinuation rate. For the active SPMS population, the presumption there was that they would remain on siponimod until the disease was no longer active. What we would like is some marker of true non-active SPMS, but what we have is EDSS health states. So in this case, we assume that patients would discontinue at EDSS 7. This is the stopping rule that has been applied for relapsing remitting models in the past and also aligns with when we see a decrease in relapse rates. So for those two reasons, we assumed that patients would discontinue at EDSS 7 and then follow the natural history course of disease. Our base case comparator is best supportive care as approximated by the placebo arm of the EXPAND trial. Disease-modifying therapies are recommended for patients with active disease. However, no DMTs have consistently demonstrated an impact on progression in SPMS. None are labeled for active and non-active SPMS. And as Ravi mentioned, we had insufficient evidence to do a network meta-analysis and directly compare siponimod to other disease-modifying therapies. We did explore a scenario with siponimod versus an active DMT based on the manufacturer-submitted MAIC. And I'll talk more about this, the methods and the results later in the scenario analysis. Efficacy is based on the EXPAND trial for siponimod versus best supportive care. And we have a hazard ratio for disability progression, which is moving to a higher EDSS health state. And we also have a relative risk of relapse. So each health state is associated with a risk of relapse, and siponimod reduces the risk of relapse within that health state. 9.4% of patients were assumed to discontinue based on what we see in the EXPAND trial. After the first 2 years, we assumed that discontinuation rates would be lower, that it would be 3% per year. And you can see the hazard ratios for the overall population and the active subpopulation, the 0.79 and the 0.67. Each health state was associated with a risk of relapse. The baseline rate of relapse in the EXPAND trial was quite low. It was 0.2 in the siponimod arm and 0.3 in the placebo arm of the EXPAND trial. We did want to retain a risk of relapse that was related to the EDSS health state. So for our overall SPMS population, we sourced those from the literature based on rates for SPMS. For the active subpopulation, we chose ones that were slightly higher. They're based on some adjusted values for SPMS. So we have two different sets. We have one for the overall SPMS population and one set that's slightly higher for the active SPMS population. And this differs from the EXPAND trial because we assumed that the patients for which siponimod might be used in clinical practice might be relapsing to a higher degree than those enrolled in the EXPAND trial, given the label of siponimod. We assume that about 71% of relapses would be mild or moderate and 29% severe. Utilities by health state were derived from a longitudinal prospective cohort study of people with MS in the UK. This was a survey where patients filled out the EQ5D, and utilities were derived based on population-based preference weights. And you can see I have a few starred. This survey provided utility values for SPMS for health states 6, 7, and 8, but not for 1 through 5 or 9. So 1 through 5, these are based on just general overall MS utility values. And for 9, no information was provided, so we assumed a utility value of 0. Other sources have provided negative utility values, so even a 0 is conservative. In addition to these health state utilities, there's a disutility associated with relapses. So 0.91 for mild moderate and 0.302 for severe relapses. Each health state is associated with a risk of mortality, which increases as your EDSS health state increases. So these are the mortality multipliers that were applied to general population mortality for each health state. Treatment costs of siponimod. So some patients, based on metabolic function, would require a lower dose of siponimod. However, both doses, the 1 and 2 milligram, calculate out to the same annual cost. Costs were based on the listed wholesale acquisition cost for siponimod. We did request a discounted net price for siponimod from the manufacturer, but they were unable to provide one thus far due to the newness of the agent. Additionally, we required each patient starting siponimod to undergo genetic screening to test for metabolic status, and about 30% to have some heightened cardiac monitoring with the first dose. In addition to drug costs, each health state was associated with direct costs of all non-drug services, inpatient, outpatient, office visits, et cetera. In a sensitivity analysis for the societal perspective, we also included indirect costs with lost workplace productivity and retirement due to multiple sclerosis. Each relapse was associated with a direct and indirect cost as well. So the relapses is on the last row there. We explored a number of scenarios. We have 2 key scenarios that I'll present. One's the modified societal perspective, so including the indirect costs associated with MS, which are substantial. Also the MAIC of siponimod versus interferon beta-1b. And we looked at a number of other things as well, inclusion of caregiver burden, which is an important consideration in MS, changing the discontinuation for active SPMS, so changing that stopping rule, maybe patients stop at 8 or 9, efficacy based on the 6-month time point, and various sources of utility and mortality values, and also the subpopulation with non-active SPMS, just to shed some light on that question as well. To elaborate on the MAIC a bit, the manufacturer submitted an MAIC comparing siponimod to other DMTs. If you're unfamiliar with this, an MAIC seeks to provide comparative evidence when traditional networks and ITCs are not deemed feasible. And what it does is match the patient-level data from the EXPAND trial with the aggregate data from comparator trials. So it will take a selection of the EXPAND trial that matches the mean age and relapse activity of the comparator trial, and then adjust for potential effect modifiers. So instead of having a network with a single network of evidence, you have siponimod compared to each individual other trial separately. We did not include this in the base case due to limitations inherent with MAIC. It's not as robust as a true head-to-head or randomized controlled trial, and limitations of the individual comparator trials. As I mentioned, the results of this are siponimod compared to each individual trial, and if each individual trial does not reflect clinical practice or a robust study, that's a limitation of that comparative analysis. And in this case, there was some inconsistency in how endpoints were defined. Clinically relevant dosing in the comparator trials, which would limit their applicability to clinical practice, and the ability to fully adjust for all differences or potential effect modifiers between the siponimod trial and the comparator trials. Nonetheless, we did want to use the information that we had, and knowing that patients with active SPMS likely are going to be treated with something. We chose one, this interferon beta 1b, which was the most similar to an active population, and maybe most similar to the indicated population of those with active disease for both siponimod and interferon beta 1b. So results. The top green here is for the overall SPMS population, and the bottom blue is for active SPMS. If you look at the third row, you see the incremental. So this is the difference between siponimod and best supportive care. There's an incremental cost of $865,000, but also an incremental gain in life years, ambulatory life years, and QALYs. But the ICERs came in quite high at 3.7 million per life year, 1.2 per ambulatory life year, and 1.15 million per QALY gained for the overall SPMS population. For the active SPMS population, the efficacy was improved, as you saw from the hazard ratios, and they also have more relapses. So there's more relapses to avert with siponimod. So we see an improved profile. The incremental cost was $407,000, incremental life years 0.26, ambulatory life years 1.24, and 0.94 QALYs gained. Nonetheless, the ICER for life years, ambulatory life years, and the cost per QALY remained well above commonly cited thresholds of $150,000 per QALY. We conducted one-way sensitivity analysis, and this is varying each individual parameter by either plus minus 10% or a reasonable range, such as the confidence interval. For both overall and active SPMS, the hazard ratio for progression for siponimod made the most impact, which makes sense, seconded by the relapse rate and the list price. We conducted probabilistic sensitivity analysis. This samples each parameter from a distribution informed by the standard error or the confidence interval. Found that you can look at the cloud for the cost effectiveness, that no iterations fell below the willingness to pay threshold for either the overall population or the active SPMS population. To summarize some of the scenarios, our first key scenario with the modified societal perspective, there was a reduction in incremental cost because there was some additional cost benefit with siponimod by reducing indirect costs as well, with no impact on QALYs. So it reduced the ICER a bit to 1.14 million for the overall SPMS population and 422 for active SPMS. So still quite above where we would consider siponimod to be cost effective. And no other scenarios resulted in an ICER that was below the 150,000 per QALY threshold. In regards to the MAIC, we looked at siponimod versus that European study of interferon beta 1b using the MAIC. About 70% of patients in this European study had relapse within 2 years, making them a fairly active population, at least by this measure. And there was a statistically significant benefit for interferon 1b in this trial. The MIC adjusted for age, EDSS, and the proportion of patients with relapse within 2 years. In running these numbers, which this was academic in confidence, so I cannot show you the individual inputs, but we found there was a reduction in the incremental cost because the comparator now had cost associated with it and a substantial cost, but also a reduction in the incremental QALYs because the comparator had a treatment benefit. Those taken together, we found there was actually, the ICER was higher than base case with the cost per QALY, and this was based on the interferon net price and the siponimod list price. As a reminder, we did ask Novartis for a net price, but it's not available yet. It's a limitation to consider. For the value-based price, we did not carry out value-based price benchmarks. The report evaluated siponimod as a treatment for SPMS, as the FDA-approved indication for siponimod for relapsing forms of MS. And active SPMS is only a portion of the patients with SPMS and does not include RRMS. We are not providing value-based price benchmarks for siponimod as part of this review. A few limitations to note. The natural history data that we used is the best available, but it is from an older study, and it might not fully reflect the current SPMS populations right now due to diagnostic and changes in treatment practices. There's very limited data on the natural history of active SPMS. And in fact, there is no data on utility values, costs, relapse rates, and efficacy of comparators specific to active SPMS. Our analysis is not reflective of the full FDA-approved label for siponimod. We started with the clinical trial and then dove into subgroups that would be reflective of the clinical trial with the active subpopulation, but does not reflect the full label of relapsing forms of MS. And finally, analyses were based on the list price for siponimod, which may not be reflective of the price out in the marketplace that payers might pay. A few comments that we received that we'd like to highlight. There were concerns raised about modeling active SPMS as defined by relapse in the 2 years prior to baseline. That is not the only nor maybe the best definition of active SPMS, but this was the predefined subgroup and aligns with the FDA language for at least one definition you could use for active SPMS. There was also concern that best supportive care might not be an appropriate comparator in the real world. As mentioned, there is insufficient direct evidence and a difficulty with performing indirect treatment comparisons. So we did what we could, and we used the MAIC as available. Relapse rates in our first draft report may have underestimated relapse rates among patients treated with siponimod in clinical practice. Given the FDA label, we think that's probably true. So we made a modification to use an alternative source of relapse rates that additionally had stratification based on EDSS. Finally, the utilities we used in the draft report contained negative utilities for some health states in EDSS 8 and 9. And a negative utility represents a health state worse than death. We received comments that there were some current concerns with these values. And although negative health states are considered methodologically sound and used previously, we did make a modification. There was a more recent source of utilities that I showed. Some limitations in that it didn't provide utility values for every single health state, and some of these sample sizes were small. Nonetheless, we updated it to use an alternative source of utility values. In conclusion, siponimod likely improves outcomes compared to best supportive care, and our model captures that. But using the current list price for siponimod, results were above commonly cited thresholds for cost effectiveness. Based on sensitivity analysis, it's unlikely to be cost effective for the overall SPMS population or the subpopulation with active disease, both for the base case and all scenarios explored. And then I'll take questions.
Don, will you kick us off? Thank you.
Yes, and I will try to be quick because I know we want to move, but I do have two questions. If you could go back to slide 51. The question I have, which is probably for the experts too, is the use of the CDP3 gets counted no matter what point in the EDSS you are, correct? And it looks to me as though in the 2, 3, and 4 levels here, there's something different going on vis-a-vis, for example, the utilities are roughly the same from 3 to 4. And then if you go to the next slide, you also see that the mortality rates in that level are close. So I'm trying to get at whether there's any expectation that where you sit as the starting point in the EDSS measuring CDP3 creates different subpopulations in terms of improvement. I'm trying to understand that.
Yeah, and before I
hope that makes sense.
Yeah, it would make a difference. So in this particular instance, about half the patients entered the model at EDSS 6, and it was clustered, right around there. So the number of patients entering the model in 3 and 4 is relatively low. But if it would have been in those states, a higher proportion, it would have made a difference to the model results.
Yeah, and I'll just add to that. It's also regarding complications of heavy immunosuppressant medications. We just presented data from Kaiser and from the country of Sweden that once they get to a 6.5 or higher, they have more infections and they have more falls. So that, I think this was one of the concerns raised with this drug, that it could make them dizzy. They see an excess amount of falling in the patients on drug versus placebo.
Thank you,
I think it's, I'm sorry. I'm just going to say, I think it's important to understand that the EDSS is not a linear model also, and some of the data that's used here fits linear models to EDSS states, but just to understand that that's now how it was developed in practice. It's transitions between, especially at the upper end, different forms of mobility aids needed and ability to get around.
So the EDSS, as was pointed out, is not a linear model, it's a categorical model. And the time at which a person stays at a certain EDSS state varies with the state. So if you look at natural history data, which goes back to the late 80s, people who entered the study at EDSS 6 or 6.5 are, under natural history conditions, likely to maintain that same EDSS for an average of about 3 years. And this was a study in which people were followed for only 18 months mean. So they're not going to pick that up.
Thank you. My other quick question was the naive non-econometrician question about the cost per life years of ambulation gained in a disease that seems like it's tending towards progression. And so I'm wondering how that relates to thinking about progression in terms of life years not lost, I guess. I don't know. Maybe that's not important to the analysis, but I was curious about why life years gained is more important than the other side.
I don't know that it's more important. I think when we were thinking about the life years of ambulation, in addition to the life year, cost per life year and cost per quality, we wanted to think about if there was an outcome measure that would be important to patients with SPMS. And given the construct that we were working with EDSS health states, that's one that we came up with that we thought might be relevant and meaningful to patients.
There's a lot that I would like to say here. I don't know if it's my turn to talk.
It is your turn.
But I understand that this is about economics, but I also would like to share my story and my family's story. My mother had MS. She was diagnosed in probably 1968. They had nothing so she went for years and years. I can't tell you how many trips we made to the hospital, how many broken bones that she had from falling, how she had to stop working. And she ended up her last couple years of her life in a nursing home because my brother and sister and I could no longer care for her at home because she was so disabled. And she died at the young age of 69. I'm 61 right now, so it scares me a little bit, but if she had had those medications, 8 months after she died, I was diagnosed with multiple sclerosis, so it was mind-boggling. But you need to know that there are people out there that need these drugs, that hope for these drugs. I'm on many organizations, I'm on many Facebook pages. People stop drugs because either they can't afford them, or they live in a very rural place. I'm very fortunate that I live in the New York metro area, but I had to give up work about 10 years ago, which broke my heart. I loved what I did in my career for 25 years, but I've changed, unfortunately. I was a very outgoing, active person. I now, since I don't work, I pretty much try and leave my Mondays and Fridays open so that Mondays I can rest up from what I did over the weekend, which wasn't much, and Fridays to rest up for the weekend, which I might. So I think you folks have to take in more than just dollars and cents and numbers here and percentages. I think you need to take in, I have non-active secondary progressive MS. I wish I could be on this drug, and maybe my physician will put me on that, but I've been on Rituxan, which is an infusion drug for the last 3 years, doing it 4 times a year. It's an off-label drug for MS. Originally had come out for blood cancers. So I fortunately have a doctor who thinks out of the box, and I always encourage people with MS to go to doctors who have a heavy MS patient population, so you're not just Mikey. But my hope here is that you folks can come to a decision that this drug is important for people with not only active but non-active MS.
Thank you. Aaron, do you have any comments?
So if I'm reading the trial correctly and how this, okay, sorry, I thought I was. If I'm reading the trial correctly, it sounds that like most people enter the model at 6, but no one in the trial was at 6. The trial was.
That's the median.
The median was 6, but most people were better.
So 6 and 6.5 is the cutoff. So 6.5 is this is the problem with the scale.
No, I agree. So it just feels like that we have a study which was somewhat flawed to begin with, and then provided limited data, and now we're applying that across the board to all the states. Are we sure that is right? I mean, does it prevent progression at these later? We just don't know, do we?
We just don't know.
But we are assuming for the point of the model that it prevents progression across the board.
We are assuming that if they remain on therapy, they'll continue to prevent progression regardless of what EDSS health state they're in.
So yeah, and that favors the drug. So to assume that it's still working after 6.5 is, biasing the analysis towards a benefit from the drug.
Harold?
I was thinking about the comments that Dr. Langer-Gould made about the trial just before. Does that change the way you're viewing this cost effectiveness analysis?
So I would say not really, because in the really big picture, this is a fingolimod Me Too drug. Novartis makes fingolimod. It's about to go off patent. We'll have generic available in the fall. It was developed initially for relapsing remitting MS because it was supposed to be the cleaned up version that does not cause bradycardia or AV block. So you could skip first dose monitoring. When they did the phase 2 trials, that was not the case. They went to the FDA, originally proposing a development program very similar to ocrelizumab, 2 identical placebo-controlled relapsing remitting MS trials, and 1 SPMS placebo-controlled trial that's almost identical to Ocrevus, where they did PPMS placebo and 2 identical relapsing remitting trials. And then they decided to put all their eggs in one basket and pull back on the relapsing remitting trials and just do this SPMS trial. When you look at those safety results, so I have some concerns. The complexity is actually not reduced with this medication. So now we say, okay, we look at the efficacy results. It's not working in an inactive SPMS population. So it's fingolimod repackaged. So now my question is, well, is there anything about this drug that makes it either safer or easier to use than fingolimod? And should we somehow maybe use it at least in some patients in our relapsing MS patients? And the answer there is no. Although not everyone needs first dose monitoring, there is a genotype that dictates whether or not and what dose you should be on, how you titrate, and whether you can be on this medication at all. And it's one of the, go back to medical school, acetylchrome P450 genes. So other drugs metabolized by the liver, they can either double or halve the levels of siponimod. If that happens, you could be at risk of having cardiotoxicity. You could increase your risk of infections and lymphopenia. And many of these medications, from a practical perspective, are drugs that we would not be prescribing as neurologists. So it's a real, when I think about, the over 200 neurologists that work in my organization, it's a real nightmare to try and figure out how you're going to make sure that patients stay on this drug safely, that they know which medications they cannot get from urgent care or from their primary care doctor if they have a little fungal infection, or if it's a woman with bacterial vaginosis. Grapefruit juice is on the list again. So it's actually more complex when we think about how we would use this in practice, and it's a lot more expensive. So I think, those things combined, I think the main reason we went back to doing this analysis and saying, well, why aren't we just pulling it and saying, because there is a lot of confusion, even though they did not really study a purely SPMS population the way they did with natalizumab, it's still being marketed in that way, and many neurologists, general neurologists, and patients still seem to think that they might have some benefit from this drug. The toxicity profile, like I said before, we looked at when you use these drugs, it's not just heterogeneity of treatment effect, it's also heterogeneity of risk. In the more advanced patients, they are going to get more complications. Seizures, there was a seizure and a fall and a fatal brain injury in this trial. The seizure risk with this drug is real, as it is with fingolimod. So the safety profile looks no better than fingolimod, yet there's no additional benefit. Now, if it's going to get promoted to use in patients that are already at higher risk of infections and falls, I have a real problem with that.
Excuse me. I just need to move on to the next part. And I want to refer the CPAC members to the packet that has some of the additional information regarding the side effect profiles. I think it's a great discussion. Lisa, fabulous presentation, a wonderful analysis by you and the ICER team.